This program is brought to you by Stanford University. Please visit us at stanford.edu.
I want to emphasize that although my name has appeared on a lot of publications, I have been spoiled by continuously having a laboratory of outstanding students and postdoctoral fellows, some of whom actually showed up tonight. So we actually have, in fact, it's interesting, we have graduate students who are in my lab, two of them. One who came from Singapore and is a Stanford graduate student, right there, Cian, and Mike Alonso, who's American-born. But then, so we have graduate students, and we have a postdoctoral fellow standing over there, Dan Weiner. And then, and there's Will Singh. I didn't know Will was here. Okay. So why they came, I can't tell you, except they're teasing me. But it was also interesting. I actually saw one of my former fellows who was living in the area, Wayne Godfrey, who showed up, and I hadn't seen Wayne in a while. And some of the people who graduate from the laboratory end up all over the world. And I've been here on the faculty since the end of 1978. I was 5 years old at the time when I joined the faculty. Gosh, wow. And since that time, I've been fortunate enough to have NIH and other grants support my research. And through the lab, a number of people have come through, and they've ended up all over the world. And Wayne, actually, I think Wayne was a postdoc in the lab probably in the 1990s, right? When did you actually leave? 97. Anyway, Wayne then spent some time in academia and ended up in biotechnology, and is now one of the senior officers of a local biotechnology company which is developing cancer vaccines, the topic of tonight's talk. So it's great to see Wayne. Anyway, and he's one of the young ones. So obviously some of the students graduated in the 1980s and are professors in various places or doing other things. In any event, I am delighted to be kicking off the effort of the Stanford Blood Center in this new approach to discussing scientific developments in sort of an informal, not so informal, here I am behind a podium, it doesn't feel very informal, environment with members of the community. And my interest, since I have started my research career here, has been in trying to understand the immune system and how best to use it for therapeutic purposes. So the immune system, I guess I don't have a pointer. Oh well, that's all right. The immune system represents our natural way of fending off mostly infectious agents. We believe the immune system plays an important role in perhaps preventing tumors, but it exists to detect and ultimately eradicate anything that invades the body that shouldn't be there. And it has many tools at its use. White blood cells and antibodies are perhaps the best known components of the immune system. And what makes the immune system so powerful and efficient is that in contrast to weapons that are not very specific, the immune system is extremely precise. The system developed in large part to be able to recognize foreign invaders, but to recognize them and eradicate them in a very specific way, call it a silver bullet if you will, such that when the immune system is successfully used, the targets are destroyed with little or no collateral damage. So that's the immune system, and we wouldn't do very well without one. There are diseases, quite a few actually, that directly affect the immune system. HIV, AIDS is one such disease, and these diseases result, if the immune system is destroyed, in the death of the host. Now, tonight's discussion is about tumors and the immune system. And there is a handout that will provide additional detail and go beyond probably what I say. And I do hope that you will interrupt me if you feel like it and ask questions, because this is supposed to be informal, and here I am providing what sounds like a very formal lecture. So the way I've described these tumors in the immune system is the way I view them. And tumors represent a unique challenge because, in contrast to most truly foreign invaders, they are not altogether foreign, because they consist mainly and sometimes exclusively of what we call normal tissue components, or self. And the immune system, as I indicated earlier, is trained and educated and fine-tuned to attack non-self and avoid attacking self. In fact, when the immune system attacks self, that creates a problem called autoimmune disease. And there are a lot of such diseases: rheumatoid arthritis, multiple sclerosis, diabetes type 1. These are all autoimmune diseases where the immune system is misdirected and, in fact, attacking self-tissues. But in the case of cancer, the challenge for us immunologists is how do we overcome the immune system's reluctance to attack self in the way of tumors, or produce tumor-specific antibodies or white blood cells that can do this. So that has been the challenge for the last, for the last 100 years. And the first approach, first successful approach to harnessing the immune system against cancer was the development of what we call monoclonal antibodies. Now, we all have antibodies in our system. Antibodies are proteins produced by a type of white blood cell called B cells. And they are proteins that are developed specifically to recognize other targets. And when they recognize those targets, they bind to them and render those targets incapable of function. We normally have hundreds of thousands or millions of individual antibodies, each recognizing a different substance in our circulation. And they're there in the event that they see something that shouldn't be there and then act accordingly. The problem is that for the most part, the antibodies against a given target are present in relatively low concentrations, and particularly low concentrations when they're directed against self components, for the reasons that we discussed before. But about 20 years ago, actually about 30 years ago, almost 30 years ago, I edited a book, a textbook, on the subject of monoclonal antibodies. And at that time, those of us involved in writing this book, predicted that someday these antibodies would turn out to be useful against cancer. And it took another 15 years from the time we wrote that book before antibodies were actually made available for the treatment of cancer. And the reason was, it turned out that it was really hard to isolate the right antibodies and produce them in huge quantities. And it wasn't until recombinant DNA techniques became available that it became possible to select out the individual gene for a given antibody, transfer that gene into a bacterial organism, grow the bacteria up, and produce huge quantities of the antibody. But that became available. In fact, a company not so far from here called Genentech, some of you have heard of Genentech, very successful company, was really the first to do this in an efficient way. And they have capitalized on that technology to be able to produce a number of antibodies which have now been approved for the treatment of cancer. And so the key to Genentech's success was, as I said, the ability to identify an antibody encoding gene and then be able to use that gene to produce huge numbers of copies of the single antibody. The other key, of course, was that the antibody has to be human in origin. Because if the antibody comes from a non-human animal, you can guess what happens when that antibody is injected into a human being. Well, it's recognized as foreign, and the immune system attacks it as foreign, and it doesn't last very long. But But it became possible to clone the genes encoding human antibodies and then produce those human antibodies in huge amounts, in pure form, directed against constituents of tumors. And those antibodies are now available, many of them, as therapeutic monoclonal antibodies. And some of them are very effective. And it happens that Genentech has several of the ones that are particularly effective against cancer. But a limitation of these antibodies is partly their advantage. And the limitation is that they only recognize, a given antibody will only recognize a single target molecule on the tumor. And in addition, the antibody has to be administered repeatedly over time because eventually it's cleared by the system. Remember, it's not being produced by the host, it's being administered exogenously, and it circulates and it does its thing for a while and then it leaves. In addition, no single human monoclonal antibody against cancer is really curative by itself. And the reason for that is because it's directed at only a single component on the tumor. And tumors can be very wily. And unless that tumor needs that target antigen or substance to survive, it will figure out a way to kind of escape from the single antibody. So antibodies by themselves today, while they're very useful and they're part of our treatment regimen for a number of different types of tumors, are generally not by themselves capable of curing a tumor.
Ed, just a quick question.
Yeah.
Have there been any case studies where the injection of these antibodies has evoked an immune response from the recipient?
Yeah, although the antibodies are human in origin, we also are capable of recognizing, in some cases, proteins produced by another person. In general, these monoclonal antibodies are incredibly safe. In fact, what makes them desirable and preferable to some chemotherapies, most chemotherapies, are relatively nonspecific, and they're designed to kill any cell that is dividing rapidly. So that could be a tumor, or it could be a cell in the GI system, because our GI cells tend to divide rapidly. So you have a lot of side effects and serious toxicities related to chemotherapies. Because the antibodies are so specific, they're generally pretty non-toxic. However, occasionally there will be what we call a hypersensitivity reaction to one of these antibodies, but it's relatively uncommon. And so compared to most forms of chemotherapy for cancer, monoclonal antibodies are exceptionally well tolerated.
What was the origin of the antibody like Herceptin, for example? Where did that first one come from? And why choose to use monoclonal rather than polyclonal if that's a weakness?
You want to repeat the question? Yeah, the question was, what was the origin of an example of one of the antibodies that's commercially available? And the one that was mentioned is Herceptin, which is used to treat certain forms of breast cancer. And then why is it advantageous to use only one antibody, or one antibody directed against a single target? Well, in answer to the first question, Herceptin was actually made and developed at Genentech. And it was made because it was recognized that the target molecule, which is called HER2/neu, is sometimes expressed or overexpressed, if you will, on certain forms of breast cancer. And in addition, the target molecule, when it functions, can actually cause the growth of the tumor. So it looked like a pretty good target. It's expressed on the surface of the cell, so it would be accessible to an antibody injected into the blood. And it turns out that when it is injected, it works to a degree. So in the end, whether or not an antibody is useful is determined by clinical trials. And in this case, you could say they were somewhere between brilliant and lucky that it worked. In about a third of cases of breast cancer overexpress the HER2/neu protein and are benefited or can be benefited from this antibody. But your second question actually is a pretty good one, and actually a sophisticated one, which is why only one antibody? Why should we only use one antibody at a time? And I think the answer to that is, well, we probably shouldn't. If we had the ability to use polyclonal therapy, I think we should. The problem is that the way these antibodies are developed and the way drugs in general are developed, your government, the FDA, pretty much requires that each candidate drug has to be tested independently and individually to determine not only how well it works, but how safe it is. And it takes, on average, for a given drug against any disease, more than 10 years before, from the time it starts into development to the time it's approved. And in the case of Herceptin, no exception. So it was in development for a decade before it was actually approved at a cost of hundreds of millions of dollars. But eventually, we believe that, in fact, this antibody is not used as a standalone therapy. In fact, it is only used in the context of being added to chemotherapy. So I think it's very likely that eventually we'll be using combinations of antibodies, just like today we use combinations of chemotherapies with antibodies. But right now we only have one that works in breast cancer, and until we have others, there won't be combinations. But it's a pretty good question. Yes?
What percentage of breast cancer tumors did you say express the HER2 protein?
About a third of the breast cancers, what we would call overexpress it, so that it's a reasonable target. And in fact, before that antibody is used clinically, it's generally required that the tumor be studied to see if it's appropriate for that kind of treatment. Okay, so anyway, that's the good and the good and the bad of antibodies. So antibodies can work; they're very, very safe, but generally speaking, they're not curative by themselves. And one of the reasons for that is because they only interact with a single molecule, and tumors generally don't need a single molecule. They can generally figure out a way to escape from a single antibody. So our approach, the approach that our group has been investigating for quite a long time, for more than a decade now, is a somewhat different approach than the use of what I would call passive antibody therapy. And the approach that we are exploring is to try to convince the patient's immune system to attack the tumor. Much more challenging, but ultimately perhaps more effective. And this requires that the old concept, an old bias, an old paradigm be wrong, which I think it is. And the old concept was you can never do what I just said, that the inability of the immune system to recognize and attack tumors is inherent and irreversible. And the reason that we thought this for so long is because our thymus gland, which is located up here and is fairly large when we're born and when we're children and gradually gets smaller, the thymus is where the T lymphocytes, a critical type of white blood cell, are trained. This is where they go to school. And every T cell makes its way through the thymus before it's released into the circulation. And while they're in the thymus, the vast majority of T cells that can recognize self are actually destroyed. So they can't get out and cause autoimmune disease. And that's how we're evolved to not all of us get autoimmune disease, because if these T cells that recognize self were not mostly eliminated, we would all have serious autoimmune disease. But it was thought that all of the T cells that recognize self are eliminated. And now we know that that's not the case. In fact, there are a lot of T cells in our circulation that can recognize self, but they're held down. They're held back through mechanisms that we don't completely understand yet. We're beginning to understand some of those. So so our challenge is to be able to identify those cells that are being held back and turn them loose, in essence, against the tumor. And the approach that we have focused on over the last 10 or more years has been to try to take advantage of a particular type of rare white blood cell called the dendritic cell. And the dendritic cells are shown here in these photomicrographs. And you can see why they're called dendritic cells because they express these long finger-like projections. And they use those projections to interact with the environment and sample the environment. And you find these cells in virtually all tissues of the body, and they're there, as I said, to sample and sound the alarm if something comes along that shouldn't be there. So in fact, here's the life cycle of a dendritic cell as we now know it and understand it. They begin in the bone marrow on the upper left, and they come out of the bone marrow as a progenitor cell. They circulate through the blood supply and eventually end up at some target tissue where they may stay for their entire dull life. And we hope that their life is dull. Here's an example of dendritic cells that are established in the skin, where we call them Langerhans cells. And they exist in the skin for the purpose of sampling the environment and hoping that nothing comes along that shouldn't be there. And they're also in the deep layers of the skin. We give them another name there. So here they are sampling the environment. And if something comes along that shouldn't be there, and we'll call these danger signals, those signals are recognized by the dendritic cells. They become activated. They develop bigger dendrites. They acquire those molecules and substances that shouldn't be there, and they make their way into the draining lymph nodes. So you've heard about lymph nodes, and the lymph nodes are those little mini organs that the immune response takes place in. So all of the key white blood cells that are geared up to attack, they're all getting together in your lymph nodes and gearing up to make an immune response. Yes, sir?
Can you give some specific examples of what the danger signals might be?
Yeah. Danger signals consist of, for example, viral and bacterial nucleic acids, RNA, DNA, and there are certain sequences, nucleic acid sequences, that are different in mammalian cells than they are in bacteria and viruses. And the dendritic cells actually have receptors on their surface and inside their cells that can specifically recognize those sequences that shouldn't be there. So that's what I mean by danger signals.
They recognize them as specifically non-human sequences?
Yes, that's correct. And there are others. There are also certain carbohydrates that are non-human. And we have a series of receptors, probably around 10 have been identified so far, most of which are in the dendritic cells. And those are geared to interact with these substances that shouldn't be there. And when they interact physically, that sets off an alarm inside these cells and turns them, they become very activated, and they leave their sites where they've been standing guard, and they exit via the lymphatics and go into the lymph nodes, where they then interact with other types of white blood cells, B cells and K cells and so forth. Yeah.
What's the lifespan of a dendritic cell, and are they constantly being trained to recognize self?
Another good question. The lifespan of a dendritic cell, we actually published some studies 3 years ago when we made the discovery in the mouse that unperturbed, as long as the skin doesn't get inflamed or infected, those cells stay there the entire life of the animal, and they never leave. They never leave. But if they're disturbed, if danger signal comes along, then they leave and they're replaced. And when they leave, they go off into the lymph nodes, they do their thing, and they die. And it's only then when they're replaced by new recruits from the bone marrow. So it's a very efficient system designed to be used only when necessary. Okay. So we actually were among the first in the world to be able to identify and isolate human dendritic cells. And in fact, one of the reasons that I joined the faculty at the Blood Center and was fortunate enough to be able to help start the Blood Center some 28 years ago was in order to be able to gain access to a lot of white blood cells. And what better location and situation to be in than to be not only working at the Blood Center, but to be the boss. So it afforded us an opportunity to access white blood cells. Now, we can't steal the white blood cells from donated blood. That would not be so good. except for the fact that we remove them anyway, because white blood cells in donated blood are actually not good for the recipients of blood transfusions. Because Larry's white blood cells, when they enter me, they don't like me because they're part of Larry's immune system, and they're going to react against me, just like mine would react against Larry's. Unless we're identical, and the chances of that are very, very small. So in fact, today, we routinely remove the white blood cells from donated blood and make them available for research, and we have for many years. And so, again, I came and helped start the Blood Center with the idea that my research would be partly for the reason that our research would be aided by having so much access to these white blood cells. And in fact, one of the first things that our group did with white blood cells was to figure out a way to identify dendritic cells and to isolate them from the blood. And it turns out that wasn't easy because they only make up about less than 1% of the white blood cells. So it was really a hard task to do, but eventually we were able to isolate these cells and study them and begin to take them apart and try to understand how they work. And we found out a long time ago that at least in a test tube, after we isolated them, they could be used to stimulate other white blood cells, and we called that vaccination in a test tube. And after several years of study, the idea occurred to me, a very simple-minded idea, in fact, that, well, what happened if we could isolate dendritic cells from patients with cancer, away from the tumor, away from all of the negative influences of the cancer, and introduce to them a substance derived from their cancer in a way that would cause them to, if we re-administered them, to induce an immune response to the cancer. Very simple-minded idea. So that's exactly what we tried to do. We used our methods to obtain these dendritic cells from patients' blood. In fact, we obtained the blood by having the patients come to our blood bank and undergo a procedure called leukapheresis. Anybody here ever undergone an apheresis procedure? Wow, that's impressive. Well, you probably underwent this procedure because we needed your platelets, right? But in fact, a very similar procedure can be performed to remove the white blood cells. And it doesn't hurt you because our white blood cells turn over so fast, and we obtain just a tiny little fraction of the white cells. But in this case, we were obtaining the white cells from the patients themselves in order to load them with tumor constituents and then return them to the patients. And so the first time we contemplated this, so here's a description of what we did. That's an example of a patient, and the patient comes into the blood center, undergoes the leukapheresis procedure, which takes about 2 hours and is quite painless. And then we obtain the white blood cells, and from those white blood cells, we purify the dendritic cells, and then we manipulate them in a test tube. We load them with an antigen or a substance derived from their tumor, and then after we've loaded them, and this takes about 2 or 3 days, we give them back. We simply inject them back into the patient with the idea that those loaded dendritic cells will make their way back to the lymph nodes and incite a revolution, if you will, and cause the other white blood cells, notably the T cells, to attack the tumor. That was the idea. Very simple concept, almost, 15 years ago. And the first time we did this was in patients with malignant lymphoma, and we had sufficient success that we were inspired to go on and try what we would call an even more challenging tumor, a type of adenocarcinoma. And we chose colon cancer, and we did a very small trial in patients with metastatic colon cancer. And then one of the patients we treated, we only treated 12 in this study. And these are called CT scans. Have you all ever seen a CT scan? It's a kind of specialized X-ray that allows you to see at one level of the body. And on the left side over here, I had a pointer I would show you. This is a metastatic lesion that shouldn't be there, here and here. And after this patient received its vaccine, the tumor went away, which is very unusual and unexpected ordinarily. So we were very excited, and we reported these results. And in fact, we did the first dendritic cell-based cancer therapy vaccine studies. And then after we reported our results, then groups around the world began exploring this approach. And now, over the last decade, there have been lots of studies done. And the results have been mixed. And it turns out that everybody's been preparing their dendritic cells using different methods. And in the meantime, of course, we've learned a great deal more about dendritic cells than we used to know. And I'll mention some of those findings in a moment. But the most advanced trial, clinical trial, because our little studies are experiments where we've studied 10 patients, 15 patients. These studies were not designed to develop a product that would be approved by the FDA for use, wide use. They were designed to determine whether it was even feasible to think about an approach like this. But eventually, other groups, even companies, have pursued this strategy. And one company called Dendrion, of which I was a founder 15 years ago, although I really haven't had any contact with them for a decade, but nonetheless, they licensed our technology from Stanford and went ahead and started developing a vaccine for the treatment of metastatic prostate cancer. And they published their results of their latest trial about a year ago, and this is what clinical trial data look like when it's published in the literature. And in essence, they divided the patients randomly into two groups, patients who were going to receive the vaccine and patients who got a placebo, standard of care, no vaccine. And the patients who received the vaccine and they were followed over the next 4 or 5 years, there was a significant difference in the lifespan of the patients who received the vaccine, who lived longer. They're the ones in brown, than the patients who didn't receive the vaccine. And that difference, even though it might look not very great here, is at least as great, if not greater, than the difference, the effect seen by Herceptin in the treatment of breast cancer or Avastin. So it's pretty dramatic. But the problem is that it was a relatively small study involving only about 150 or 160 patients. And the FDA said, this is a great study, but we want to see more data before this product is approved. So we're waiting to find out if their current clinical trial is as successful as this one, in which case then perhaps this will be the first dendritic cell therapeutic vaccine to get approval and be used. Now, in the years that have followed since we first started working with these experimental vaccines, one thing has become clear, and that is that they're incredibly expensive. In fact, when we did the first study, when we first started treating patients in the early 1990s, it cost us about $500 or $600 per patient to prepare the dendritic cells. Today, if we were to try to do that first trial, it would cost us about $30,000 to prepare the same cells. And the reason for that is that the FDA has stepped in and said, we want to regulate this whole cellular therapy approach to make sure that it's safe for patients, but the regulatory requirements are such that it's much more expensive. So now, every time we would want to do a study like this, we would have to prove and test and test again that the preparations are pure, that they don't have any infectious agents in them, and that they're reproducibly consistent of the same constituents. All things that seem at the surface like very reasonable expectations. But the problem is that the tests that are needed to do that sort of quality assurance are very expensive. So it's virtually impossible today to do the same studies that we did 10 years ago to show that this actually works. So I think it's unlikely that you're going to see a lot of cell-based therapies of this type in the future because they're simply too expensive to develop and ultimately too expensive to use clinically. Now, having said that, I don't want to rule out the possibility that Dendrion's prostate cancer vaccine might ultimately get approved. But just remember that each time the vaccine is made, it has to be made from the patient. It's not like you have one vaccine that treats everybody. So each vaccine is unique for the individual patient, And that's what makes it so expensive. Yeah.
Is it possible to just isolate only dendritic cells from other people, or does that go along with the fact that you can't use white blood cells in someone else?
Yeah, unfortunately, your dendritic cells would be recognized by my T cells. In fact, efficiently recognized, because dendritic cells are so powerful at presenting antigens, they would be rejected very quickly. So no, that won't work. So that's a real limitation of this kind of individualized therapy.
How do you identify an antigen specific only to the tumor?
Pretty tough. The question was, how do you identify an antigen that is only expressed by the tumor or specific to the tumor? And as I said at the very beginning, tumors consist mainly of self-constituents. So most tumors, as far as we know, don't have unique antigens that are not expressed on other cells. In fact, one of the risks of taking a monoclonal antibody is that the target of the antibody is often expressed by other cell types. It turns out, however, that tumors are more sensitive to the effects of the antibodies. It turns out that tumors are more sensitive to the effects of the vaccines. But there will be some collateral damage. Now, so far with antibodies and these vaccines and others like them, we've seen relatively little collateral damage. There is, however, some collateral damage, and in fact, one of the approaches to cancer immunotherapy that is getting close to approval is an approach that does result in considerable autoimmune disease. But in general, when you're treating patients who have diseases that are going to result in their death, the alternative, having an autoimmune disease that is treatable, is currently viewed as okay. So it's an interesting point for discussion, but yes?
When you have people that are metastatic and some of them are in the placebo group, should we assume that they've already exhausted every kind of approved and experimental form of chemotherapy, or do they just have to sign saying, you know, I want to be a part of this and I'm aware that I'm making a placebo group?
Yeah. Okay, the question actually is a really good question, and that is when patients participate in a clinical trial and they're assigned to either receive a placebo or the real drug, how can that be? I mean, I think one of the questions you're saying is that doesn't sound right. But the problem we have is that how do you know objectively when something works or it doesn't work? And it's been found over many years of trial and error that the best way to determine whether a drug works is to not have the patient know what they're getting and not have the doctor know what he or she is giving. And that's called a randomized, placebo-controlled, blinded clinical trial. So that's the best way to find out the information. Now, if the patient gets the placebo and the drug really works, that's not such a good thing. So from an ethics perspective, this is a topic of endless debate and discussion. And some trials, particularly with life-threatening diseases, in fact, the first AIDS trials with drugs to treat AIDS were not done with a placebo control. And if something works so spectacularly that virtually everyone who gets treated, or the majority of patients who get treated, get cured, you really don't need a placebo control. But unfortunately, most of the new drugs that we develop are improvements. They're incremental improvements, but they're not generally dramatic cures. And so for those kinds of drugs, you really need these controlled, well-controlled clinical trials to find out whether they work or not. Unfortunately, they're very, very expensive. And as I mentioned before, the average cost to develop a drug from the time of conception to the time of approval is estimated at between $500 million and $1 billion. That includes some of the, I think that overall cost includes some of the drugs that have failed, but nonetheless, the average cost, when you average in the ones that have made it and the ones that haven't, is just enormously expensive, which makes it pretty rare when you get a drug that makes it across the finish line. It just takes so long and it's so expensive. But that wasn't why you asked the question. You were asking about what happens with the patients. Now, the other point I would add is that today, most clinical trials of patients who are who have cancer, when the drug candidate is first tested, it's initially tested in patients who have advanced disease. And the reason for that is, again, an ethical argument that we'd like to give those patients a chance, and at the same time, what have they got to lose? Whereas if you treat a patient who has earlier stage disease, for which there are other treatments that might be curative, it's felt that it's inappropriate to expose those patients to unproven drugs. Larry?
But those patients are probably undergoing other treatments at the same time.
Yeah.
Test. And do you worry that there's interference?
We absolutely do. The question was, what effect might there have of other treatments that patients are receiving on top of the new drug? Or I should say, how do you discriminate or distinguish the effects of the new drug from all the other drugs that the patients are getting? Well, that's part of the clinical trial. So let's say all the patients are getting standard therapy, which could include 3 different chemotherapies, and then you add on top of that the experimental drug or the placebo. So the patient isn't getting nothing as a general rule. Now, there are some tumors for which we don't have meaningful treatments, but for the most part, most patients in this country are getting other forms of therapy, and you add on to that therapy the new therapy, and then you compare the outcome. So that's how it's generally done. Yes?
Could we, in general terms, discuss the relationship of drug prices to the cost of developing and the cost of producing them?
Wow. Well, that question has to do with the cost of development and the cost of producing and the relationship between the pricing. The U.S. is the only country in the world where there is a semi-free market when it comes to pricing. Now, in point of fact, that pricing is modified by the government. Medicare and Medicaid negotiate pricing. But as a general rule, I'll give you a general guideline because these rules are not written down anywhere. As a general rule, drugs that have dramatic benefit for life-threatening diseases are generally priced higher than drugs that are Me Too's or have only modest benefit. That's a general guideline.
Independent of their cost of production?
Independent of their cost of production. Now, monoclonal antibodies, in fact, any recombinant DNA produced protein is very, very expensive to make. Genentech probably has spent billions of dollars on their factories to produce their antibodies. And then they charge as much as they possibly can for their antibodies. Their antibodies are really expensive. What is their profit margin on when they're ultimately sold? Very high. But right now, society says they deserve the profits because their drugs are so effective. Genentech stubbed its toe in the past year because they, in one very interesting story, because Genentech is actually a company to be admired. Look what they've accomplished. But on the other hand, they developed a monoclonal antibody to treat colon cancer and lung cancer called Avastin. Some of you may have heard of Avastin. And it's moderately effective. And then they used the same basic antibody to target a disease called AMD in the back of the eye that can cause blindness. And it turns out that the target protein, which is called vascular endothelial growth factor, VEGF, not only causes cancer cells to grow, but it causes the cells in the back of the eye to grow and ultimately causes blindness. But they made a different form of the antibody called a Fab fragment to inject in the back of the eye to treat the eye disease. And they developed it as a separate drug with a separate price. And they charged a whole lot of money for that drug. And some ophthalmologists got a hold of the original Avastin drug, which is used to treat systemic cancer and used in much higher doses. And it turns out they could buy a vial of Avastin for, let's say, $2,000. And they divided the Avastin into about 100 little vials, each of which they could treat back of the eye disease. And Genentech became very angry and very upset. In fact, to this day, there's a big fight going on because Genentech argues that Avastin, their drug, was only approved to treat cancer. It's not approved to treat AMD in the back of the eye. And so they don't like the fact that their drug is being used inappropriately to treat a different disease probably because they can't make as much money. So yes, there is a certain amount of capitalism, if you will, and to suggest that pharmaceutical companies are not motivated by profits would be untrue. Even Genentech, that has been so successful and so innovative. So these drugs are very expensive, and they tend to be more expensive in the U.S. than they are elsewhere because we don't have the same level of price controls as there are outside of the U.S. So it's a topic that we can talk about for a very long time, and there are pluses and minuses to the arguments. But William.
Is there a long turnaround time on making an individual vaccine, or is it a matter of a day or?
Yeah, well, for the kind of stuff that I described, the turnaround time is about a month. Now, when we did the original experiments, it was 2 days. And the reason it's a month is because the FDA requires that you test the vaccine to make sure it's sterile, and that takes a month because you have to send it off for fungal tests and viral tests and bacterial tests. And by the way, in all of our vaccine studies, we never had a positive test. But that's the requirement. So in fact, it takes several weeks for that kind of quality assurance, quality control. Yeah, Mindy.
Can you expand in patients' dendritic cells?
Yeah. Yeah, you can. The question was, is there a way to actually expand the dendritic cell population and perhaps freeze them away so that you can use them again and again? And yeah, the answer is yes, you absolutely can. There are growth factors that we've used to expand the number of dendritic cells, and there are other ways to do that. You know, what I didn't mention to you, and I should, and maybe I have a slide here, but I'm not sure I do, is that the approach that we're taking, I don't mean to sound pessimistic about cancer vaccines. I am pessimistic about the use of manipulated dendritic cells because it's so expensive. But what we're doing in the lab now, and we've been focusing our research on, is to come up with ways that can actually target the dendritic cells that are already in the patient without having to remove them. And in fact, we and others have identified drugs or drug candidates that can actually get to the dendritic cells and activate them without having to remove them and manipulate them. And this is much more potentially much more efficient, much more cost-effective, and pharmaceutical companies would love this approach. So this is actually the future of dendritic cell-based therapies, in my opinion. And in fact, there are a number of such drugs that are now in clinical trials, and I apologize for not getting to that point sooner, that look very promising. And as I mentioned before, certain of these drugs appear to activate the immune system so efficiently that they do create some autoimmune disease in the process. So far, that has proven as an acceptable side effect. I think we're going to know within the next 6 months or a year at most whether one or more of these new drugs will make it across the finish line and get approved by the FDA. But I think it's pretty clear we're on the dawn of a new era. The monoclonal antibody approach is here to stay. There will be more and more monoclonal antibodies that come through clinical development and are approved by the FDA. But I think we're going to hear more and more about these newer approaches that are designed to turn on the patient's immune system to attack the tumor. They won't be what we started out with, this kind of primitive, very expensive dendritic cell manipulation ex vivo and give the cells back. I think it's going to move more toward manipulating the cells with small molecule drugs, but have the same effect. So it's actually a very promising time for us, and I think over the next, probably 5 years or so, you'll hear about these drugs as they make it through the phase 2 and phase 3 clinical trials. So we're in the middle of a great time from the point of view of understanding the immune system and using the immune system to fight cancer. And so I'm very optimistic. So I didn't mean to be pessimistic about the approach that we initially took. And this is an out-of-date photograph of the people in my lab. You can see a predominance of women here. That has actually evolved. Right, Mike? He's very unhappy about that. And Dan? So now it's about 50-50. But most of these are young, they're all younger than I am, young graduate students or postdoctoral fellows who are here at Stanford for anywhere from 2 years to 5 or 6 years doing this kind of work, and then they go on and do hopefully great work when they leave here. They're a very happy bunch, and you can see them standing around our Stanford Blood Center sign, because without the Blood Center, this kind of work would not have been possible. And I've already mentioned some of the ways. I mentioned the fact that we get our dendritic cells for research from white blood cells that would otherwise be thrown into the garbage. The patients undergo their leukapheresis procedures here. None of these newer approaches to cancer would have been possible here at Stanford without the fact that we have a blood center that not only has a conventional blood bank, but also interacts so closely with the teaching and research activities of the university. So I think with that I'll stop, and if there are any other questions, we can continue the discussion. One other thing that I should mention to you, and that is the idea of prophylactic vaccines against cancer. Now, when I use the term vaccine here tonight, I'm talking about therapeutic vaccination. Traditional vaccines are designed to prevent diseases, and everyone here has been vaccinated against one or another infectious agent, whether it's hepatitis or influenza or tetanus. You've all been vaccinated, I suspect, to one or more of those. And the way that works is, and this has been going on for many years, is we've been able to identify parts of the virus or the bacterium that are harmless, and we inject those parts, or we kill the whole virus in the case of certain viruses, and we inject those killed organisms or pieces of them, and the immune system reacts, and we are forever protected because the immune system has what we call memory. I go into that in your handout. You can read about it a little bit. And that is the basis for every prophylactic or preventative vaccine that we have today. It's the immune system. We're fooling the immune system. The immune system thinks that your body has been infected by this horrific pathogen, a virus, and it's reacting to it as if the live one were there. And because it has memory, if you're ever exposed to the live virus or the bacterium, the immune system will react immediately and hopefully beat it down before it has a chance to get a foothold. Now, in the last 2 years, the first prophylactic vaccine against cancer has been developed and approved by the FDA. Do you know which one that is? I know some of you probably know. It's a vaccine against cervical cancer. Cervical cancer. And one was developed by Merck, and another one was developed by, I think, Glaxo, two mega-sized pharmaceutical companies. And the way that vaccine works is it turns out that cervical cancer is caused by a specific virus called the human papillomavirus, a sexually transmitted virus. Most people who are infected never develop cancer, but a percentage of these infected women will over a period of many years. In fact, that's why women undergo pap smear screening, because there is a gradual change in the appearance and morphology of the cervix that predates the actual cancer. And if you catch it in time, you can treat the lesion surgically, and cancer won't develop. Well, in parts of the world where routine screening is not performed, cervical cancer is actually very common. It's less so in this country. But because the cancer is caused by a specific virus, it was possible to take components of that virus that are in and of themselves harmless, like any other vaccine, and inject them into people, healthy young people, induce an immune response and an immunological memory, and over a period of about 10 years of study, the sponsors of those vaccines, Merck and Glaxo, were able to demonstrate that their vaccines prevented cervical cancer because they prevented infection by the papillomavirus. And today those vaccines are available, and it creates an interesting dilemma, because who are supposed to be, who are the targets of those vaccines? These are generally healthy, very young, preteen or teenage girls. And an argument rages to this day whether all such young women should be vaccinated or not. It's a very interesting philosophical question. There's no doubt that the vaccine will prevent cervical cancer. Absolutely no doubt whatsoever. Will it prevent 100%? No. But it's probably somewhere between 80 and 100% effective. And as far as we know, there are no serious side effects to the vaccine. So it's going to be very interesting to see how ultimately this discussion, this argument over whether and who should be vaccinated with this effective cancer vaccine will come out. But that's the way it is today. It's the only cancer vaccine. There will be others that are developed that are prophylactic, but this is the first one.
The preceding program is copyrighted by Stanford University. Please visit us at stanford.edu.